This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 556, recorded on July 12th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
What's with the fluttering eyelashes?
No, I'm just glad to be back, that's all.
Yeah, the last few times you were in Fort Lee.
I was, or AWOL.
And for 3 weeks you've been fishing.
Yeah, and sightseeing and things like that.
Although we haven't actually recorded here every one of the last 3 weeks. You know, we've been on the road and so forth.
Yep.
But welcome back. Do you have a good fishing trip?
Had a great time.
All right.
Wonderful.
Wyoming, I understand.
Yeah, southwestern Wyoming. I stayed at the town of Kemmerer, which is noted because it contains the mother store for Jcpenney. Mm-hmm. And it eventually, I think, will be the only Jcpenney in existence the way it's going because all the other stores are closing.
Yeah, I thought they were out of business.
No, they're not. No, they're not.
Other parts of the country.
I think about 50% of the originals are not in business anymore.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. And it's also really warm here.
Even
How
inside?
warm is it?
Actually, inside it's quite cold, but I have to wear a sweatshirt inside because they really like to air condition my office.
Yeah, we do that.
Do you have Drew University on your sweatshirt?
I do have Drew.
How did I know?
Sweatshirt.
How did I know? If I sent you a TWiV sweatshirt, would you wear that?
I would. I actually have a TWiV sweatshirt.
Oh, okay.
I'm not wearing it today.
29 Celsius here in New York City.
29. Well.
Here as well. And very sunny.
And humid.
And very humid as well.
Yeah.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here, it's 73 Fahrenheit.
Oh, my gosh.
A bit chilly.
Yeah, I guess that'd be around, I don't know, 22.
Yeah, could be.
Yeah.
Something like that.
22, 23 could be. Yeah. Yeah, the Celsius is compressed. That's why people don't like it.
Yeah.
Too bad. Right? And from Western Massachusetts, Alan Dove.
Good to be here. It's 32 Celsius here, 90 Fahrenheit, a bright summer day, and the storms have cleared out. And by the way, you mentioned Jcpenney. Can you name a famous virologist who worked at Jcpenney?
No, I can't.
I can't.
Maurice Hilleman.
Really?
Yeah, you know that because you interviewed him.
That was his first Very job. Cool. His aspiration was to enter management at Jcpenney, and then somebody suggested he ought to go into science.
Oh.
Darn.
So, if he had stayed at Jcpenney, they probably wouldn't be going out of business now.
That's right.
Yeah, we wouldn't have.
But I think we're better off.
We wouldn't have a lot of vaccines that we have, right?
Yeah.
That's right.
Made a lot of vaccines or directed their production, right?
Maurice Hilleman was or is still, I don't know if he's still alive or not.
He's gone.
He had a reputation of coarse language.
Oh, very coarse.
Yeah.
Very coarse.
Oh, very.
Oh, yeah.
And at Jcpenney, that just wouldn't have flown.
It's a Midwestern company, right? Jcpenney?
Yeah, well, I told you where it's from. No, it's
Wyoming
not Midwestern.
Is not Midwestern.
Western.
Western company?
Far Western, in fact.
All right.
It's almost in Utah.
Well, speaking of passing away, Kathy, you want to tell us about?
Yeah, the news this week is that Bill Yaclik passed away. So Bill Yaclik was the founding president of ASV. It was really his idea and activities that got it going. And the ASV meeting next week, it was already set to honor him and his accomplishments. There'll be slides about him, a poster about him. The keynote address is the Yaclik address. And some family members were going to attend, but now I'm pretty sure that will not be the case. But we'll have a lot of aspects about ASV history at various points throughout the meeting. But his accomplishments are just amazing. I've got a link here to the obituary online. You can also get to it from the ASV website, where we always have an in memoriam section. And That's about all I'm prepared to say at the moment.
Was he working when he died, or has he been retired for quite a while?
He's been retired. Yeah.
Because I remember when I was in graduate school, I was at Notre Dame, and of course Maurice
Green.
Morris Pollard.
Morris Pollard
knew him very well and spoke about him very glowingly. Mm-hmm. Where was Yaclik employed again? Remind me.
At Duke.
Okay.
That in memoriam site on the ASV website. That's not where you want to be, Dickson.
No.
I was looking at it the other day and said, "Well, I'll be there one day, I guess."
I guess.
Hopefully, it's a long day in the future.
If they choose to do that.
Yeah, they may not want to put Racaniello on the page. It's too long a name.
Dr. R.
Well, we tend to put members when we find out about it, and we also sometimes have non-members there too.
Yeah, well, it's
about this time, I would say support us. Go to microbe.tv/contribute. We actually have two little letters that actually say it better than I can.
Oh.
The first from Mark. Hi, Twivers. No weather, just a simple haiku for you.
Yeah, that's a good one.
Racaniello shares science knowledge for free. Help him with some funds.
Wow.
Enjoy your
I think it has
summer.
to be Racaniello.
That's right.
Yeah.
After the five syllable.
Yeah, it doesn't actually have 5 syllables, but we can make an exception.
Right.
Enjoy your summer, Mark. And then Folka writes, Dear Twix listeners and fans, this is a letter to you. There must be many of us enjoying the science and banter that Vincent and the team bring to us at least once a week. But if you look at Patreon, only 200 of us have already decided to say thank you by giving a small amount each month. Are you one of those many listeners who really like Twix? Are you fortunate enough that you can easily afford a cup of coffee? Maybe you already considered becoming a Patreon. Then please press pause right now and do it. It's really easy. It will not reduce the number of coffees you will drink. It will make a real difference to the Twix team. Just imagine if Vincent and the others would only consider doing the podcasts, but not actually do them. Thank you. Greetings from Climate Change Hot Germany.
Climate change.
Folka.
That is interesting.
Yes, thank you both for writing that. It's very nice of you.
Yes,
Well
very
put.
good.
And microbe.tv/contribute if you want to help us out.
Unless if you're driving, don't drive into an abutment or embankment or
Right.
whatever you might drive into.
Please.
Now, you might notice that Brianne sounds different today.
Brianne, why do you sound different today?
I have a new microphone and a new headset.
Well,
Very nice.
I sound really good. Someone asked me at ASM, why does Brianne sound different from everyone else? I said, oh, because she has a headset, and I've been meaning, and that reminded me I had to get her. I've been promising a while to get her.
Yes, and you all sound very good too.
Yeah, the headphones help too. So they're
They
comfortable.
do.
Everything is good.
Very comfortable. Everything's very good.
And you have a boom attached to your desk, right? That you can move the position.
wow.
I do. I have a boom attached to my desk. Everything looks nice. Only a couple people asked if I was setting up a karaoke bar in my office.
Yeah, now we all have good microphones. So
That's the
it should
idea.
all sound good, except when Rich is traveling or something. Then
I presume he's in Bend, Oregon right now.
he's in Europe, actually.
Oh, is he really?
The last pictures he sent us, he sent me a picture from the Louvre the other day.
Nice.
And before that, he sent pictures in Bath, England.
Yep.
The
Been there.
pool, which is very green.
I've been there. That's a Roman bath, by the way. And
Yeah,
it was
I know.
the site of an outbreak of acanthamoeba.
I would expect it to have been the site of an outbreak of a lot of things from the pictures.
Yeah.
Well, they drained it as a result, and then they discovered all these little mementos that the wives of Roman soldiers had thrown in, wishing them luck as they ventured out into the wilderness of Scotland. So
I was there
quite
a number of years ago, and I remember in the town there was a restaurant where they had glasses of the spring water.
really
You could say, but it was hot. The water was hot, yes.
put a tea bag in it.
Not particularly interesting to drink.
No.
So I think he will be back next time we record, which will be in 2 weeks. Well, we're going to record on next Monday at ASV, actually.
All right.
And he will be there. Although, of course, next Sunday, we will release an episode. We do not fail to release episodes.
Well, that's not a we thing. That's you. And we all tip our hats to you and our capsids.
Yes.
We tip our capsids to you.
You.
You tip your capsids.
We do.
I like that. That's good. Sometimes I wonder if I'm just too driven.
No, I think you're passionate. That's the best way to put it. I've never met anyone so devoted to a science. Well, maybe a couple of more people like the Duve or something like that. But you are a shining example of the ways a person devotes their life to the science that they grew up with.
I do have fun, as you say. I have fun when I start podcasting in particular.
Right.
And we all do. That's why we do this. Okay, so we have a snippet in a paper today for you. We will try to
We may actually be able to do the snippet as a snippet.
Yeah, this one is a snippet, actually.
It's pretty fast.
Well, this is a very interesting letter in Nature Medicine called Delayed Childhood Neurodevelopment and the Neurosensory Alterations in the Second Year of Life in a Prospective Cohort of Zika Virus-Exposed Children. First author, Karen Nielsen-Saenz, and the last author, Maria Elizabeth Moreira. And this comes from, the main study is on a cohort in Brazil, but we have people from UCLA. We have Oswaldo Cruz. You were just mentioning that, Dickson. Biomedical Research Institute of Southern California, São Paulo, University of São Paulo, Medical University of Graz, Austria, Göttingen Medical Center, UCSF,
Sweden,
Beijing. Wow.
But all the cases, I presume, came from Brazil.
Yes.
This is a cohort in Brazil.
This cohort is all in Rio.
Yeah.
All right. So Zika virus, of course, caused an epidemic in 2015, 2016, which was first noted in Brazil.
TWiV.
TWiV. That's right. And it became well known because it was causing microcephaly in some children born to mothers who were infected during pregnancy. And the question has always been, does it do anything else? So the children are born with these obvious skull defects.
Some of them are.
Some of them, not every one of them.
Yeah.
As you'll see today, we actually have a number. But what else can happen? If they're born without any obvious structural defects, do other things happen? So that's what they address in this paper.
So these were only children that were exposed in utero. Is that the idea?
Yes.
Right. These are
Not
children
later,
born
after they were born.
to mothers who were RT-PCR positive for Zika virus infection. They were symptomatic and RT-PCR positive.
That's a good question, though.
But something is after you're born, nothing bad happens?
Well, I don't know if that's necessarily true, but, brain development, the wiring continues for a while, right?
Right.
Yep.
Is the development completed at birth, but the wiring now goes on for a number of years?
It has to, because you're making more neurons to fill up that.
So I think that's a really interesting question, but that's not what they address here. Right. This is a cohort where the mothers were PCR positive, as Alan said.
But no obvious,
had a rash.
Physical defects that you could detect in the birth, so they assumed.
Well, some of these kids had microcephaly.
Some.
8 of 216. That's 3.7%. Had microcephaly.
Right. So as the epidemic was unfolding, they enrolled 244 pregnant symptomatic women.
And then they followed them up.
Which
is what this study is.
That's a difficult study.
It's hard to do. And they said some people dropped out and didn't do everything, but they got quite a number.
And one of the things that they weren't able to get was a negative control.
That's right.
Yes.
That's right.
I was, as I was reading the paper, I was like, well, how does this compare to the rate of your control group? And they talk about that a little bit.
But again, remember, this is as we'll see, they compare it against a scale.
Yes.
It's like a growth. You bring your child whose weight, and they compare it against the scale of hundreds and hundreds of kids. So in a sense, it's being compared against that scale. But I agree that, and they even say that they should have a control, but that's hard to do. Yeah. So first, 8 of the 216 infants live. So there were 223 live births, and they ended up following up with 216. 8 of those had microcephaly.
And in fact, 4 of them had it initially, and then 2 of them subsequently
got better.
And 2 of them, and 2 of them got better, but 2 of them got it after birth, I think. Anyway.
Yes.
Yeah. So
and one of them had it surgically repaired.
Right.
Developed normal head circumference following surgery. So it's interesting. And I didn't know that it would reverse, but apparently that's not all the kids are reversing, right? It's just a few.
Right. Microcephaly is determined based on a score, like head circumference especially.
Yeah.
And so you could score a kid, okay, they've got a smaller than normal head and they therefore meet criteria for microcephaly. But then because everybody is an individual, they happen to develop at a rate that allows them to catch up. And so they're no longer microcephalic.
Yeah.
So the other question that I would also like the answer to, and I'm sure you know, is that microcephaly, is it restricted because of the bone of the skull rather than the actual growth rate of the brain inside? Or if you relieve the pressure from that by surgery, will the brain just grow normal?
No, the problem occurs during development.
Understood.
The brain is not
the whole brain and head do not develop to the proper size, and the bones are not fused at birth, so you could have the brain continue growing.
No, I knew that, but I'm sorry that I didn't explain my question better. So why did you think these two that had it got better?
I don't know.
Well,
I know is that the brain is smaller and that the skull is therefore smaller, right?
So it adapts to a smaller brain rather
it's
than
not
the other way.
The skull is not restricting the brain size at all.
Right, no.
Okay.
So 216 of these infants, they had the follow-up, and this is between 7 and 32 months of age. And they had clinical and neurological evaluations. So they had hearing tests, they had eye exams, and then the neurological evaluations are done by the Bailey scales, which are a standard set of ways to evaluate things like cognitive development, language, and motor skills.
APGAR didn't figure into this. No, really?
Well, that's a score that's done right after birth. This Bailey assessment is done during development.
But Bailey was a person. She developed these. And APGAR was here, right?
Sure.
Yeah, she was here.
Absolutely.
And so, all right, so hearing and eyes, and then cognitive, language, and motor. Those are the areas that they're addressing here. Mm-hmm. And they also had some questionnaires and neurological examinations that were done. So 71.3% of the children had normal assessments. 59 of 146. That's 40% had these Bailey 3 results less than one standard deviation and in at least one of these 3 areas that I mentioned before. 40% had a problem with either cognitive language or motor.
More than a standard deviation below the mean.
So that's below average.
Yes. So another interjection, since I didn't read the snippet, and I'm sorry to say I didn't, but I can be the
you don't have to admit it. You
can no, it shows in my questioning. Of all these kids that were followed, were they all, did you say, I'm sorry, I must have missed the point. Did you say they were all seropositive at birth?
All of their mothers.
The mothers.
So how can we be sure that the fetus was infected or not?
Well, that's a great question. We don't know that. We're just looking at what happened to the kids. And, I think this is a higher than normal number having these issues at this time of life. Although you would like to have a control group. I totally agree.
To have a control group.
No, understood.
But these are below normal for sure in terms of normal Bailey scales.
But not all
so
of
this is as good a place as any to talk about the control group. I mean, the Bailey scale is presumably based on some generalized population, and these are often done in Western rich countries where you set the standard for it. So now you go to Rio and you're finding mothers who were infected with Zika virus during pregnancy. I'm just going to hazard a guess that you're somewhere to the lower end of the socioeconomic scale if the mothers were infected with Zika virus during pregnancy.
Perhaps.
Right? Because, I mean, rich people are going to be living in air conditioning there, right?
Right.
But wasn't there an athletic event that
there? Right. So it's not that you couldn't be exposed if you were wealthy and privileged, but it seems like you'd probably be less likely. So the question is, among a corrected cohort or a matched cohort that's demographically similar on things like income and education level and maternal age and all those, what would be typical Bailey scores? And the answer is we don't know.
Right.
But there is a piece of there are some suggestive data in here that imply that what's going on with this group is significantly Zika associated.
They do say in one part that statistically significant associations with maternal age, education, mode of delivery, infant birth weight, and hearing function were not seen.
Right.
But they don't sort of talk about those things on the Bailey scale.
Right. That's correlating developmental issues with any of those things in this cohort, but we don't have the comparison cohort.
Yeah. They just say that figure 2, they would have expected would look like a bell curve.
Yes.
And here they see it being shifted to the right.
That was one of the things. So the whole thing is shifted. Yeah. Instead of seeing, they see basically none of these kids are way above average. Mm-hmm. And a significant number of them are below average, and that your bell curve has been shifted toward the below average end. And I think we're probably going to talk about this in a moment, that there is a risk factor, significant odds ratio of developmental problems associated with earlier infection during the pregnancy, right?
I think the question of whether they're all infected is really interesting. So this bell curve shift, it could be a combination. Maybe they're all infected, and that's a consequence, even in ones that don't have these overt issues. Or maybe it's a reflection of the population. We don't know.
Can't you look for Igm antibody in the newborn?
Well, they could have done that, yeah.
Didn't they take
or
serum?
Any antibody.
Did they take serum? No, not any antibody, because the maternal antibody is IgG. So the child's antibody would be IgM.
Would be M.
So they could have looked in the serum of these newborns to see whether they were infected or not because of this IgM antibody. Like toxoplasma.
They didn't report it, although they could have, I suppose.
Yeah.
Right, because they're following these individuals up for...
Sure.
Everything they've done here is non-invasive, right?
Right.
Yeah.
Right.
So if they had found only IgG and the ones that were normal or semi-normal, that might imply that that's a protection against the infection for the newborn. From the mother.
Yeah.
The same logic applies to toxo and lots of other
I want something explained. So if a mother is infected during pregnancy, she will initially develop IgM.
Which doesn't cross the placenta.
It will not cross. So at what point will she develop IgG?
Probably 3 or 4 weeks after she's infected.
Okay.
That's about what I would say as
well.
So
she is passing IgG to the baby.
Well, it depends on the trimester that you're talking about.
Yeah, of course. Okay. And then in turn, the child, but then it would be too late to protect the fetus from infection. Right.
Right. Exactly.
Yeah.
Okay. No, those are all very good questions. Yeah.
So one other point that they sort of make at the end, having to do with the control group, is they felt like they couldn't get a control group because so many people in the area were infected with Zika.
Yes.
Yeah.
And there were, people who were maybe asymptomatic but still infected.
Yes.
I know that this would be very difficult to do, but it would be really interesting to see if moms with asymptomatic infection also had babies with some of these same issues.
Yeah, that's a good question that they raise. If symptomatic versus asymptomatic, is there a difference?
It seems to me that using this Bailey scale to evaluate newborns might have been applied to other epidemiological studies not related to Zika in the past when Zika was not even in Brazil. So you could use that data in retrospect, perhaps.
Maybe.
Yeah, I don't know where else you would look for a cohort like this, right?
Right.
I mean, certainly not in the U.S.
No.
Now, if it turns out that the Bailey scores are mostly from Western nations, then it's kind of not fair.
Right, but I think actually Dickson's suggestion is excellent. To look back at the historical record, if maybe somebody used, now this Bailey score has evolved over time. They're using the Bailey 3, but if you could go back several years and maybe somebody did an analysis and that would give you that figure.
Exactly. Some sociologist, perhaps, or some psychologist, or whatever.
Or some obstetric study.
Yeah, that's right.
Mm-hmm. So, of these scores, Bailey scores, language was the most affected. 35% of children had scores below 1 standard deviation, followed by cognitive. No, so cognitive were 9.6 and motor 16.4. So language was the most affected.
Indicating that various areas of the brain are being affected. So it's not.
Yeah. And then the eye exams were abnormal in 9 of 137 infants, 7%. And we know that Zika virus replicates in the eye.
Right.
And so that's not surprising. Hearing deficits in 13 of 114 infants, that's 12%.
Did they rule out any other transplacental infections that might have occurred during the same time?
That's a good question, and they didn't mention
so,
it at
like
all.
toxoplasmosis is a big issue with lots of people. And
in Brazil?
Everywhere.
Everywhere.
So it could have had an effect that's similar to what you're seeing here as well. It could have masked.
Do you think they should have screened for it?
Sure, you
Now,
bet.
by antibodies?
Yeah, to look for all the organisms that are common in their environment that are transplacental.
So it could confound these results.
It could. It could.
It's interesting.
Yep.
Okay. Now, there's a wonderful figure here which shows that all these women are infected throughout gestation, starting at week 3 all the way to week 38.
Right. So they were viremic all the way through.
Well, they're not. No, this is when their infection was identified.
Eyes.
That's what I mean to say. So you have people infected in the first, and the second, and the third.
Raise another question. Right. How long is a woman viremic that's pregnant during her pregnancy? So what is the window of opportunity to transfer the infection?
What I was going to say is, and as Alan mentioned, an earlier gestational age is a significant predictor of below average neurodevelopment. So if you get infected early on, you're more likely to have problems.
Yeah, that's right.
And that is, of course, because the bulk of brain development happens earlier. Right by the third trimester, bulk of it is done, and so you're going to have less of an effect on these
right.
things.
And that actually gets to this control group issue that we've been harping on. If this was just a general societal issue that you always expect these lower than average Bailey scores in that population, you wouldn't see any correlation with timing of onset of Zika virus. So that really, I think, is an important piece of data that points to this being, a lot of this at least, being Zika related.
So would Toxo show a similar effect?
Yeah, and so is rubella.
Depending on when you're infected.
Correct.
Yes?
Yeah.
So earlier is worse?
So let's put it this way. When the placenta is like a little, maybe the size of a silver dollar during early development, the chances of it being infected are very low because the area of circulation is not very large. It gives time for the mother to develop immunity. But if the child does become infected during that time, it's tragic. I mean, it results in a stillbirth usually. The second trimester, the placenta is much larger. The chances of becoming infected are much higher if she becomes infected during that time. But because the brain has undergone development, like you were saying, then the result is harder to interpret because it's gray zone rather than black and white. And in the third trimester, when the mother becomes infected, let's say with toxo, the child that's born usually doesn't exhibit any symptoms because their brain development is fully complete, mostly by the end of the third trimester. So while the chances of becoming infected are greater, the chances of being affected are low. And that's also dependent upon maternal antibody as well. It's complicated, but I had to talk about this to medical students for 38 years, and and I had to put it on the board and say, look, it's a crapshoot. You can't advise your patient to abort just because they became infected during this particular time or this particular time.
Right.
So it's up to individuals to choose.
Now, here's some interesting things here. So 49% of the children with abnormal findings early in infancy, in a previous study they had done, were normal in the second or third years of life.
That's interesting.
And some that were normal at birth became abnormal later. 25% developed neurodevelopmental issues later. So in other words, you can't tell what's happening at birth.
Right.
You don't know it. So, and that's why they say we have to follow up all of these Zika infected kids or kids born to Zika infected mothers. And as Brianne said, they have to be symptomatic or asymptomatic, both, because we don't know if there's a difference. And they say initially they say, they should be followed for 3 years, twice a year, neurodevelopmental, ophthalmologic, auditory evaluations. Right. But then they say, who knows if it's going to, these may also affect learning, in which case we should go out to 7 years.
Yeah.
Because hearing and eye issues could affect learning, and so we should really look at them until 7 years.
And they saw stuff that was not, just the microcephaly that we've been talking about so much. I mean, the autism spectrum disorder, couple of kids, and that kind of thing. So you'd need to follow them in a way that would allow you to detect additional problems.
Yeah.
And I mean, they only picked. Oh, sorry. Go ahead.
No, I was just gonna say I just wondered why they picked 7 years. To go out to 7 years.
Yeah, I think because school age, that at that point they're established in school, and hopefully people are assessing them as they progress in school.
Why not 6? Why not 10, 12, whatever?
Right,
it just seemed very precise.
Why not 5? Why 7?
That was my question.
Sure. And by the way, this study assumes that the parents were normal with regards to these issues and that their children became compromised due to the infection rather than some genetic aspect. So it's very difficult to pick out the underlying normal without knowing another group that was similar.
Of course.
That's the biggest reason I would say. This is worthy of a letter.
Well, they say a limitation of the studies. We did not have a control, and as we said, it would be hard to do in this population. So it does have limitations, but I think it's still worth following up these kids wherever they are to make sure there are no issues, because you don't want to just let them not be taken care of, right?
Right.
But so the interesting thing here, despite the reservations, let's assume that a number of these issues are caused by Zika virus infection. We all thought microcephaly was the thing, but that's a small part of it.
Right.
There's way more that's happening, as we can see here. And as we said, if the kid gets infected after birth, what's going to happen then?
Right.
We don't know these things.
And this demonstrates that as these kids grow and develop, things change. Some of the ones that were microcephalic are not, and some of the ones that didn't have any problems at birth develop them, and that's why this needs to be followed.
So maybe there is the opportunity. Is Zika still being transmitted in Brazil?
Sure.
Of course it is. So I think that they should probably redo this study and get that control group under her.
I was looking at the numbers yesterday. South America, 2018. Okay. There were, what was the number? Between 10 and 20,000 suspected cases, and only 1,300 were PCR confirmed.
Right.
So it's still a lot of transmission. South America is the biggest epicenter still of transmission. It's not as big as it was in 2015, but it's still happening.
Allow me to inject the fact that Redi, R-E-D-I, an Italian monumental contributor to science in general, invented the concept of the control. And that was a long time ago. And we're still going to that and saying that's the gold standard. If you don't have that control, then you can speculate all you want, but you're really not sure of what you're speculating about.
So in this case, this paper was allowed to be published because, and this is my speculation.
Go on.
First, it's Zika.
Yeah.
And it seems you can almost publish anything these days. And the second is they had these charts, which is kind of an average of a population, although, it may not be the right population.
And you know what Redi said when he was learning English, don't you? He says, if you Zika, you will find her.
Yes. But the publication of this is very important because this is a unique cohort and they were actually able to follow it. And I'm impressed that they lost so few to follow up.
Yeah.
It is. These are good data to have out there, and I don't think they were given particular slack on getting it out.
Zika 1.0. I'm looking forward to 2.0.
Yes.
There was an article this week in the New York Times entitled "The Zika virus is still a threat. Here's what the experts know." And when I started reading today's paper and noticed the first author, I was happy because I recognized the name of somebody who is quoted in the New York Times article. So the first author, Karen Nielsen, or as she's listed on the paper, Karen Nielsen-Sains, was quoted in the New York Times. And one of the things at the end of the New York Times article is that while there's possibilities for vaccines, it's kind of the same situation as Ebola, that right now we're at a trough or a dip, and there's not enough cases to really figure out whether a vaccine is going to work.
Until the next epidemic.
Well,
Right.
There's enough in the DRC. It's just that they can't properly vaccinate.
True.
With a good vaccine, you wouldn't have to worry about more people justifying it. They'd be gone.
When I was in Rotterdam in May, I spoke with a lady who was actually organized the Ebola vaccine trials in Western Africa. And she was the senior author on the paper that we discussed, actually. And she said the vaccine will be licensed any day now. Okay.
Well, no,
it was in May, so it's still not licensed.
Many days have elapsed.
Which surprised me, but she said that. But yes, it's hard to test them. But with thousands in South America in 2018, it seems like you could if you had the right population. And, but they do have to go through phase 1 first.
Sure.
So
right.
A lot of them are still going through that. Mm-hmm. We have a paper. This is a article in Cell Reports, which is open access. N-glycolyl-neuraminic acid as a receptor for influenza A viruses. We have Frederick Brosite. Do we have co-first authors? Yes, what we got here? I don't know. Someone tell me.
So. No, I don't think so.
And the last author is Robert de Vries. And it is from Utrecht University, Scripps Research Institute, Amsterdam Medical Center, University of Georgia, Icahn School of Medicine at Mount Sinai, Hokkaido University in Japan. And I heard about this at the meeting I was at in Baltimore a couple of weeks ago, which was a Center for Excellence in Research and Influenza Surveillance or something like that, one of these government acronyms. And there was a lot of excitement about this paper, so I thought we would talk about it. Mm-hmm. And so, of course, everyone, I think every person on the planet knows that Cyalic acid is the receptor for influenza, right? I know, not everyone on the planet, but it is the receptor. So Cyalic acid is a sugar. Indeed. It is a 9-carbon monosaccharide, and it's typically the last sugar in a chain of sugars attached to a protein, what we call glycoproteins, right? Cyalic acid, as Dickson and I were saying earlier, it's the penultimate. It's the ultimate, but often linked to galactose as the penultimate sugar, right, Dickson?
Yes.
Penultimate.
Yes.
And so the linkage between cyalic acid and galactose can be different chemically. It can be what we call alpha-2,3 or alpha-2,6.
Right.
And I will not quiz you on this. It's okay. But just take it for a word that it's a different chemical linkage. And it's important because different influenza viruses will bind to cyalic acid depending on what that linkage is distal to the actual sugar. It's kind of interesting.
Well, lock and key situation.
Loch Ness? I'm kidding. So, for example, the human influenza viruses that infect us, they bind to alpha-2,6 linked cyalic acids, whereas the avian influenza viruses bind to alpha-2,3 linked cyalic acids. Are we okay so far, everyone out there? I know we lose you easily. Now, this is important because...
And we're going to get pretty far into the weeds here, but this is an open access paper, so if you want to grind through it on your own, you can do that.
We have alpha-2,6 linked cyalic acids throughout our upper respiratory tract. It's only when you get into our lower tracts do you find alpha-2,3, although you do find it in the eye.
Oh, boy.
And so some avian influenza viruses can initiate infection in the human eye, and that's been seen in poultry workers.
Seen? Did you actually
observed?
Say that?
It's been observed in poultry workers, and the virus can replicate, these avian influenza viruses can replicate in the eye, and then they will drain into your, there's a duct that goes from your eye to the, the upper nasal tract, of lacrimal duct, I think it's called. Mm-hmm. The virus can then get in, and then it can infect your lower lungs. So that's one way. But mostly the human viruses infect us efficiently because our upper tract, like our nasopharynx, has lots of alpha-2,6.
And droplet infection is common.
Now, when avian viruses infect people and get transmitted, they often change receptor specificity to be able to bind alpha-2,6.
Or don't you mean that the ones that have already changed were selected for?
Yeah, they're selected to be able to.
That's right. Because
and that's the fear with H5N1, which is highly pathogenic, is that it will change its receptor specificity and cause horrible outbreaks. But that hasn't happened yet. So this is all about this sialic acid, which now we have to do a little chemistry. Sialic acid is this 9-carbon sugar, and another name for sialic acid is N-glycolyl-neuraminic acid. Okay, there's an acetyl or acetyl group. I'll call it acetyl group connected to one of the carbons. N-acetyl, and that's here. What it is, it's a nitrogen bound to a carbon, and then there's a double bond oxygen. Now, it can be what they find in this paper is a related sugar, N-glycolyl-neuraminic acid, where we have an extra hydroxyl on the acetyl group, and that makes it glycolyl, okay? And so that's going to be a focus of this.
So you can think of two forms. So there's sialic acid, alpha-2,3 linked or alpha-2,6 linked. Within both of those, you can have N-acetyl or N-glycolyl modification
that's
on the
right.
end.
And that's at the 5th carbon, if you're interested in the detailed chemistry, the C5 position. Now, putting that hydroxyl on there, that's called hydroxylation, right?
Indeed.
Is done by an enzyme. And humans don't have it. So we don't have any glycolyl-neuraminic
We don't have any
acid.
N-glycolyl sialic acid.
We don't have it. And other animals don't have it, including dogs and ferrets and seals. They don't have it. And those are all hosts of influenza viruses. But horses and pigs do have this enzyme, so they can have N-glycolyl-neuraminic acid. And in fact, they do. All right.
But they also have N-acetyl.
They have both. That's right.
They have both forms. So they're so horses and pigs have both forms. Humans and ferrets and seals and dogs have only the one form, the N-acetyl.
Right. Good so far, Dickson?
Yep.
All right. So that's the little background there. And this paper, they were interested in. All right. Let me give you a little more information on glycolyl because it's relevant here. Okay. So most of the influenza viruses out there, as far as we know, are binding to N-acetyl-neuraminic acid or N-acetyl or sialic acid. That's the name for N-acetyl-neuraminic acid. Okay. There is of one virus of horses. It's called an Equine H7N7 virus. It binds specifically to N-glycolyl-neuraminic acid. But it binds to it. It's not clear whether it's using that to get into cells. And a H5N1 mutant has been produced in the laboratory that has a single amino acid change, and this interacts only with N-glycolyl-neuraminic acid. And that virus replicates very well in cell culture, even though it doesn't bind N-acetyl-neuraminic acid, right? Mm-hmm. So that's an artificially made virus. And as you will see in the end, there doesn't seem to be anything out there in terms of influenza viruses that's currently circulating, as far as we know, that exclusively uses N-glycolyl-neuraminic acid.
Right. This H7N7 in horses that used N-glycolyl was in an outbreak and then, I guess, burned out and is not circulating now?
Mm-hmm.
Yeah, I think... Yeah, that's right. It's not circulating anymore. It was highly pathogenic and burned out. So it's not around anymore. So, there are lots of influenza viruses out there. We haven't looked at them all, but of the ones we've looked at, none of them seem to exclusively bind N-glycolyl.
Right. So
up until now, all these studies looking at N-glycolyl versus N-acetyl have been using either some kind of a cell system or some slightly different sugars rather than the actual oligosaccharide structures that we're talking about. So they use some surrogate system, and so these authors are now going to just do pure, straight up biochemistry hardcore.
Yeah, they wanted to know what's the story between N-acetyl and N-glycolyl.
Right?
Can we?
And let's do this with just the pure sugars.
So
in fact, in the discussion, they described that the previous studies were elegant.
Yes.
Right. I like that.
And they used brain-specific gangliosides.
That's right.
And as Alan said here, they're going to just use the chemicals.
Yeah, it was weird. They said they were elegant, but then they criticized them.
Then they criticized.
I don't think they criticized
them first,
them.
And then they later said they were elegant.
Yeah, they said.
No, I don't think they criticized them. They just said, this is how it was done previously with, probably the reagents that they had at hand and so forth.
Exactly.
And now we have this ability to do this very complicated chemistry that they spend most of the materials and methods on instead of on other things that I would have found useful.
So
But
just to put it in context, because that's what Rich Condit would do. He's not here. He said, without being able to bind to a host cell through a receptor, you're not going to get inside that cell.
No. That's the end of the line for you.
It's done.
You need to bind. You need to get in, at least for influenza viruses.
That's right.
Now, a very important tool they use here is called a glycan microarray.
Right. I liked that.
This has been around for a while. I've heard of these many years. They have made their way into virology, but they're basically, you can synthesize many, many different kinds of sugars. In this case, they took derivatives of N-glycolyl-neuraminic acid and put probably hundreds of derivatives. And what they do is they have a robot that spots them onto glass slides in a very regular array. And so now, if you can imagine a glass microscope slide, for example, with many small spots, each containing a slightly different sugar, in this case, derivatives of, these N-glycolyl-neuraminic acids. And then you can ask what will bind to them. You can devise assays, fluorescence assays, for example, and ask what binds. So that's what they did here. They made a big chemical library. I don't know actually how many different compounds they did here. With derivatives, and there's a figure here where they show you how, they took each kind of neuraminic acid and did various permutations on them. They put them on the glass slides, and then they add initially purified hemagglutinin proteins, which are the viral proteins that are binding the cyalic acids. They call this printing when they put the sugars onto the. Now, I wanted to ask the TWiV crew here, if you look in the materials and methods, they talk about the chemistry, and they use the word how we installed this onto the molecule. And I've never heard that used in chemistry. Installed, yeah. Right?
Right.
So I'm not a chemist, so maybe I just didn't know that. But
maybe that's what the chemistry kids say these days. I don't know.
I have never heard that from one of my chemistry colleagues.
General procedure for the installation of 2,3-linked neuraminic acid.
Sounds like they went in there with wrenches and hammers. Yeah.
So many years ago, when David Baltimore was made president of Rockefeller University, I went to the
installation.
That's right. And one of the speakers was Sidney Brenner.
Oh, yeah.
And he's a funny guy, or he was.
Yeah.
He's no longer funny.
That's right.
He died a few months ago.
Indeed.
And he got up and he said...
He might be hilarious. You don't know.
Yes, wherever he is. He said, when I first got the letter that I would speak at David's installation, I thought I would bring pipes and wrenches and assemble something here. He was a funny guy, right? I would install something. So that brought that to mind. Okay, so what they did is they had these glycan arrays, and they made different hemagglutinin proteins from a whole bunch of different viruses. And for example, they have an H5 from a Vietnam strain that binds to alpha 2,3-linked N-acetyl-neuraminic acid, so that's kind of a positive control. They have the Equine H3N8HA, to see if it would bind N-glycolyl. So, and they go through a whole bunch of different HAs. So basically, they're making these proteins, putting them on the glass slides, and then I think they use antibodies to detect the binding of the proteins to the sugars. Right.
Either antibodies or another reagent that's specifically a protein binding, a lectin that binds to carbohydrates.
Right. A lectin, like the carbohydrate binding proteins.
Exactly.
And so they can say, do the viruses that we know bind, and do we find anything else interesting? And they tested a whole range of wild type proteins, pandemic, seasonal, human, swine, avian, dog, different subtypes. none of the proteins showed any responsiveness with N-glycolyl structures on the array, and exclusively bound N-acetyl-containing glycans. And this one mutant, the H5Y161A, it does bind the N-glycolyl sugars on the slides. And they say that the this acetyl and glycolyl specificity is exclusive. You do one or the other.
They looked at the horse one too, the H7.
Yeah.
Right.
That
and
binds
that one
also the glycolyl.
That binds the glycolyl, but it does not bind the acetyl.
That's right.
Right.
So I think that's the conclusion from that. You can find very few that will bind N-glycolyl. Most of them bind N-acetyl.
And there's nothing that they can find that binds both.
Nothing binds both, at least when we're using this particular assay, right?
Yes.
But as you'll see, when you use virus, it's slightly not clear, I think, right? Mm-hmm. Now, the next thing they did is to look at the structure of the HA to see if we can understand what allows for N-glycolyl to bind instead of cyalic acid, right? And we have had structures of the HA for years, and we have structures of HA bound to cyalic acid. And so they solve structures of a variety of HAs, including this H5 protein with the mutation that makes it specific for N-glycolyl. That's what you'd like to know. How is it that this can be? And they see differences, right? There are specific amino acids and loops that are known to interact with cyalic acid, and they're clearly different. So they say that this Y161 change, they call it a mutation, but of course it's a change. It's Y161A. Just being pedantic. The mutation and the consequent conformational changes and disorder is what mediates the shift to N-glycolyl specificity. So that's very nice. Of course, it would have to be something that changes the specificity, and here you could actually see it. They also looked at the equine HA, the H7, which can bind N-glycolyl to see, and they see similarity in the ribosome, in the receptor binding site residues, so that these two HAs would both bind N-glycolyl. Similar things are happening in the receptor binding site. They weren't able to get the N-glycolyl in there with the crystal structure, which would have been nice, but it's okay. All right, now we have a couple of interesting experiments, and let's see if I can summarize these in a good way. So this virus, this H5N1...
If not, you can summarize them in a bad way.
I'll summarize them in a bad way. Someone could correct me. H5 virus with a Y161A change, single amino acid, which makes it now exclusively bind N-glycolyl-neuraminic acid, right? They say it had diminished plaque forming ability, which I don't know, I guess it means small plaques, right? And in the process of growing up that virus, they found there was a second amino acid change in the HA, T160A. But apparently this has no effect on receptor specificity. It doesn't change it in any way. So this Y161A HA will still bind N-glycolyl-neuraminic acid on its own. Alpha 2,3 linked N-glycolyl, which makes sense because this is an avian. It's an H5 hemagglutinin. Okay?
Right. Okay.
Yes.
Clear.
Now,
Crystal.
And this is also on MDCK cells, which are from dogs.
Yeah, so MDCK from dogs, which of course do not have the N-glycolyl.
Right.
They're N-glycolyl negative, and they confirm that. But they find that N-glycolyl specific viruses can infect MDCK cells. So this H5 virus with the Y161A will infect MDCK cells. So it must be, there are two possibilities, that there's a little bit of binding of N-acetyl-neuraminic acid by that virus, or it could be that the neuraminidase is actually mediating binding. There's a paper published a while ago, which we actually covered here on TWiV, showing that neuraminidase, of course, binds cyalic acid. That's part of its function, and it can mediate entry. So maybe that's what's going on here.
What was the ratio of viral particles to cells affected to rule out the possibility that just resting on the top of a cell is enough to allow it to be engulfed? And once that happens, then
viruses never rest. They're always moving. I don't think that just random collisions can explain infection. It's my understanding. You got to bind.
No, but why is the plaque so small? They occurred. The fact is that they occurred.
They don't actually explore.
You're going to have to do that.
The second mutation, by the way, the second amino acid change restores the plaque size. That's why they got interested in it, but it didn't have anything to do with receptor binding.
But tell me why even the small plaques shouldn't have been there based on the chemistry.
Was this in MDCK cells? Does anyone read? They don't actually. Yeah.
Yeah.
Yeah, it's in MDCK cells.
I mean, on the glass slides, it was exclusive.
Well, it must be that.
Right.
So these are N-glycolyl specific viruses, and MDCKs don't have any N-glycolyl. So
Understood.
It must be that they're small because the viruses are entering via either neuraminidase or Cyalic N-acetyl-neuraminic acid. It's inefficient because there's not much of it, so the plaques are small.
It's inefficient.
Now, why the second?
You could prove that by just eliminating
small,
that
or
with
are they
chemical.
Are they just defective in plaque formation?
Yeah,
was it?
That's a good question. But their idea was maybe the second amino acid change was changing receptor specificity, but it's not.
Right.
So they went through this whole thing, and so in the end, they don't know why these viruses are able to infect MDCK cells.
The biochemist would say, well, what's the Km? What is the binding constant, right? And the binding constant increases as the specificity increases. And so does this have a low binding constant or a high binding constant? I mean, I would just...
I don't know, but you know what experiment I would do? You could knock out the Cyalic acid enzymes in these MDCK cells and see if that prevents infection.
Sure.
And it probably would because there's no N-glycolyl-neuraminic acid.
Right.
So there's no other way. So, but it doesn't tell you if it's neuraminidase binding or hemagglutinin binding.
Or you could flood it with the reduced sugar and see if you can block it that way by exclusion.
Oh. Which sugar? N-acetyl-neuraminic acid?
Yeah.
You could. So let me summarize this. So this H5 virus with a single amino acid change in the HA, this is supposedly specifically binding N-glycolyl-neuraminic acid, but it will infect cells that do not have N-glycolyl-neuraminic acid.
So there must be something else going on.
Well, it's Cyalic acid, and it's either HA or neuraminidase binding.
Easy to prove, right? Just cleave off the Cyalic acid and see what happens.
You know, that should prevent infection, yes. But
100%.
They didn't do that. It should.
Mm-hmm.
From the mouths of babes.
Now, next experiment. Here's an interesting experiment. Hemagglutinin binds Cyalic acid. The other glycoprotein on the virus, the neuraminidase, has to be able to cleave it off the surface of cells. Otherwise, new viruses that are made would never leave cells. They would stick to the surface. So there has to be a hemagglutinin neuraminidase balance. In fact,
and that's sort of how Tamiflu works.
Exactly. Tamiflu inhibits neuraminidase, so the viruses that are made never get off the surface. They stick to receptors as soon as they're made, right? So they say here, this Vietnam H5N1, the neuraminidase must be able to cleave both N-acetyl and N-glycolyl-neuraminic acid from cells because this virus can infect productively. So it must be that there's a balance. So they ask, can neuraminidase cleave both? They make recombinant neuraminidase, and the answer is yes. So that's the other interesting thing here. Normally, neuraminidase cleaves N-acetylsialic acid from the second sugar, which is galactose.
The penultimate sugar.
Apparently, if it's N-glycolyl-neuraminic acid, the neuraminidase will still recognize it.
Right.
And that's important because it's the same neuraminidase in these viruses, whether they're binding N-acetyl or N-glycolyl-neuraminic acid. So that it's important to show that the neuraminidase works, and it does. It works for both. They did some other experiments with inhibitors, but the bottom line is that we conclude that NA can cleave N-glycolyl, maintaining an HA-NA balance if the HA binds N-glycolyl to get into cells. Okay? How about tissues? Wouldn't you want to know what's going on in tissues?
I would.
Sure.
So they said we looked at binding of HA proteins to canine and equine tracheal tissue sections.
There you go.
Right? The canine doesn't have N-glycolyl.
Right.
The equine does. For H5 wild type, H3 equine, there's binding to both canine and equine because they're specific for alpha-2,3 linked N-acetyl-neurominic acid. That makes sense. For the H5 mutant and the H7HA, they only bind to equine tracheal tissues.
Yep.
It's because they only have N-glycolyl-neuraminic acid. So that's important because trachea is, of course, one of the places where the virus is replicating. If you move out of cell culture into tissue sections, you see that there is N-glycolyl binding by these two HAs, these two unusual HAs, right? One is just made in the lab and one which is from this weird outbreak.
Right.
So that's can determine species infection by these viruses, N-glycolyl binding. So that's the paper. It is some influenza viruses can bind N-glycolyl-neuraminic acid and infect perfectly well. It's restricted to species, certain species. And, they have a nice picture of how this is happening on the virus particle. Now, the one virus, the H5, is a mutant made in the laboratory. Okay, so you could say, well, that's irrelevant. But then the others is equine H7N7 virus, which only binds alpha-2,3 linked N-glycolyl-neuraminic acids. And they say it's tempting to speculate that this specificity led to the virus being restricted to horses, but we don't know if that's the case.
Right.
Avian H7N7s bind alpha-2,3 linked cyalic acid are still circulating, right? Because they're binding cyalic acid, alpha-2 linked acetyl and acetyl-neurominic acid.
Yeah, and they make this point that there seems to be selection for acetyl binding.
Yeah.
Given that there is no glycolyl virus circulating. So you might wonder, well, why isn't there some big horse and pig epidemic using this glycolyl binding virus? Why have those completely gone away?
Yeah, no virus circulates in nature that is known to bind N-glycolyl. So that's something is working against it, right?
Right.
Where do they say about this horse?
Oh, last paragraph of the discussion. This can be rationalized because the natural bird reservoir does not express N-glycolyl, and the equine H7N7 virus was highly pathogenic but not maintained in horses.
They killed him too fast.
So we don't really know where this H7N7 virus came from. I guess it showed up in horses and wasn't maintained.
Yeah, it's gone.
Right. But it seems as though being able to replicate in birds is an important aspect for these flu viruses.
Right, for sure. And then in birds, it's N-acetyl-neurominic acid. So I guess the bottom line here is that, every flu virus that we know about is binding to N-acetyl-neurominic acid, but you can identify a few that will bind N-glycolyl-neurominic acid. The infection works in culture. And why we don't see more of those is really an interesting question, right? That's the bottom line. Now, after the talk on this at the flu meeting, Peter Palese got up and he was all excited because he said this has huge implications for Tamiflu use. But having now read the paper, it doesn't actually, because the neuraminidase can cleave N-glycolyl-neuraminic acid from cells, so it doesn't matter. Tamiflu will still work on viruses that bind N-glycolyl-neuraminic acid. In fact, they do an experiment, right, with the neuraminidase showing that a neuraminidase inhibitor blocks cleavage of N-glycolyl. So Peter, it's no problem. Don't worry.
Yeah, the implications, they've settled a couple of issues in the biochemistry of this receptor, which is important to do, but in terms of medical and public health implications, I'm not sure that there really are any.
No, but it's the interesting question is why don't we see more of these viruses circulating?
Well, no, the simple answer to why you don't see more of these circulating is because they can't infect birds.
Exactly.
Right.
Would be my guess.
Yeah, I would agree.
So Vincent got to be pedantic earlier, and I want to do it now. I would love the authors to explain to me, well, first of all, they never define the abbreviation RFU, but I've decided it's relative fluorescence units. And
that's what I
in
thought
the
too.
last figure, they show it with an arrow pointing down on the y-axis. And maybe that's because it's supposed to point to the other panels of the figure, but really it wouldn't have taken up any more space. But in earlier figures, it was most confusing because it didn't even have that kind of appearance. And I spent an inordinate amount of time trying to figure out, why does the RFU arrow point down on the y-axis
when the numbers are going up?
When the numbers are going up. And of course, when I was reading it, I had to pull up my magnifying glass because I'm reading on a hard copy, and the light's dim. And so, yeah, I don't understand those downward arrows at all.
Yeah, it's weird.
End of story.
In fact, the legend is supposed to explain what's going on in the figure.
Yeah.
And they don't always.
Well, you can blame the reviewers for that.
Well, for example, these graphs where they're looking at binding of HAs to, or viruses, I guess, in this case, to the glycan array, the figure legend says evaluation of receptor specificity.
Yeah, or verification of the microarray control lectins.
But what
they
did you do?
They don't tell you we're doing a binding assay.
Yeah, exactly.
And the RFU thing, the only thing I can think is, for whatever reason, they put the abbreviation RFU near the top of the axis, and then the arrow, if you go from there down, the numbers go down. But it just looks weird.
Yeah, it does. And the only way I got to RFU was in the methods at one point when they talk about this assay. They say, after staining, the slides were scanned at the appropriate excitation wavelength and images are analyzed.
Yep.
Yes.
And so I was like, oh, well, then there's fluorescence.
Yes.
Yeah.
But they never define RFU in the paper, right?
Right.
No. And they don't tell you that they're doing a fluorescence assay on this array.
Well, that's Dickson. It's a reviewer issue.
It is.
Either a reviewer didn't pick it up, or if they mentioned it, they didn't bother to change it.
Maybe they just thought that's where the authors went to college.
I've never seen an arrow put on a graph like that. That's pretty weird. Anyway, but folks, if you're writing papers, make them clear. It's better for everybody.
Tell me about it.
Make it clear.
And then give them to like the most junior person in the lab and have them tell you what they don't understand.
That's right.
Because they will be great canaries in the coal mine for you and tell you what's not clear about the paper. Yep. As a reviewer, it drives me crazy when I feel like I've spent more time analyzing the paper than anyone who wrote the paper.
How about a grant reviewer? It's even worse.
Yeah.
All right, let's do a couple of emails. We have an update on measles from Jimmy. Let's see what we have here. The Sacramento Bee. 90 Sacramento area schools don't meet vaccine guidelines. Are they a tinder of a disease wildfire?
I'm seeing a lot of signs, printed and then stuck in the soil next to a shopping center.
In Wyoming?
No, here, Fort Lee.
Did you see anything in Wyoming?
I saw a lot of things in Wyoming.
No,
I didn't see any
with
measles.
respect to vaccine stuff, no?
No, I met a few anti-vaxxers, if that's what you mean.
You did?
Yeah, I did.
Did you get in an argument?
No.
Okay.
No, we were talking about fly fishing.
Rich writes, guys, many thanks for another thought-provoking episode. The discussion on Hep D reminded me of the distinction between viroids and virusoids. I think that Hep D is similar to a virusoid as it is encapsulated by a helper virus, and viroids are not. Coming back to the plasmid discussion in an earlier TWiV, but would appreciate your thoughts and also whether these are recognized terms. I also remember trying to clarify the difference in a virology textbook and could not find these terms referred to, so it may be worth covering these in the next edition of Principles of Virology if this is not already covered. Thanks again. So a viroid is certainly a recognized term. A viroid is a small RNA, circular RNA, which codes for nothing, yet it can infect plants and cause disease. And this was originally coined by T.O. Diener when he first discovered them in the 60s, because they didn't seem to be anything like viruses, so he called them viroids. Virusoids was used a little bit in papers, but it never stuck, and it might have been applied to Hep D because Hep D is kind of like a viroid, but it doesn't code a single protein, right? So someone called them a virusoid, but I look at Hep D as a virus.
Yeah.
Right? It's a virus. Hepatitis D virus. We used to call it a satellite, but I don't think we need to call it a satellite. And as I said, insult it. It's a virus. So Virusoid, I'm not going to put in the textbook because I don't think
it's the viral version of a plunet.
Ooh.
Is that Pluto?
That's this new. I think that's the new term.
The moons that circulate the large planets.
It's moons that escape their planets,
or that escape.
Plunets. Okay.
That's
become plunets.
Plunets.
Anyway,
Right.
Does everyone agree on Virusoid? Do you hear it used very much, or
No?
This letter was the first I'd heard it used.
Yeah, I've seen it in some papers I've read over old papers, but it didn't catch on, and so I don't think it's a good thing.
It sounds like something you'd call someone.
I mean, even a viroid. I mean, they're viruses. They just because they don't code for anything doesn't mean they're not a virus. But viroid is there and it's stuck, and so we use that. But Virusoid never did. Okay. Alan, can you take the next one, please?
Sure. Anandi writes, "Hi, Vincent. My name is Anandi, and I've been an enthusiastic listener of TWiV since I started my master's program at the National Institute of Virology in India, and then continued to do a PhD in adenovirology at Umeå University, Sweden, under Nicholas Arnberg's supervision. It's so exciting that you will be recording a podcast at our annual virology symposium in Smogen. I recently started a postdoc in Gothenburg in cell biology and will miss the meeting at Smogen this year, which is so unfortunate, but still exciting nevertheless. Looking forward to that episode. Best wishes from a gray and rainy Gothenburg."
All right. I'll be there. Do you know Nicholas, Kathy Spindler?
I sure do.
Okay. Yep. He's been arranging my trip.
Cool.
He'll be
Nice.
there.
Yeah. Good guy.
Dickson.
Anthony writes, "Death as a problem for biology is one of TWiV's."
In one of the TWiVs.
You're wondering why biology, in quotes, evolution, never solved the problem of death. For biology, death isn't a problem. If someone's 30 years old and has progeny that can reproduce, as far as evolution is concerned, there is no need for them to be around anymore. If someone's 30 years old and has no children, there never was a reason for them to be around at all. Since life is a vehicle for the transportation of information through time, the question isn't why organisms die after reproducing. The problem for investigation is why individuals persist after that all. My speculation is that in an inverse of altruism, if species live in groups and learning is important to all, there will be a collective advantage to maintain those with experience, even in species not in groups. As a quick glance, learning is associated with longevity. Mice are instinct-driven and have a lifespan measured in months. Bats learn and live for years. And the information-generating life is just a collection of typos. We can hope that it's more than that, believe that it's something else, but we can't think that there's meaning. AO, and then he gives a quote from Shakespeare, which reads, should I read it?
No, it's okay.
Okay. "Life's but a walking shadow." I think Alan should read this because you've got the Shakespearean voice. Go for it, Alan.
"Life's but a walking shadow, a poor player that struts and frets his hour upon the stage and then is heard no more. It is a tale told by an idiot, full of sound and fury, signifying nothing."
Well read. Well read.
It's a great one, isn't it?
It's fabulous.
It's from Macbeth.
Did we really wonder why biology never solved the problem? Because you don't want to solve the problem.
Ask
of death,
a bristlecone
right?
pine tree that question.
No, everything would pile up.
No.
That's exactly right. You have to recycle everything, for God's sakes. That's what it's about, recycle.
I mean, most organisms are optimized to reproduce, and then after that, it doesn't really matter.
That's right.
Right, and it matters. I mean, as Anthony says, it matters if the post-reproductive ones or the non-reproductive ones are helping others survive, especially in a group setting. Then they do matter a lot, and there's a reason for them to stick around. But in species that just are all about the reproduction, like rodents, then once you make the next batch, you're done.
I mean, look at the lifespan of a salmon or a mayfly. Those are great examples of organisms that just do one thing. They grow up and they reproduce, and that's it.
Justin sends us a link to an interesting article. Rotavirus vaccines may lower kids' chances of getting type 1 diabetes. And he writes, "If this does decrease type 1, I wonder what the anti-vax logic will be to deny it." So that's a study published, a study of Australian children published in JAMA Pediatrics.
Yeah, they analyzed private insurance data for close to 1.5 million U.S. children who were infants at the time of enrollment. 41% reduction in the incidence of type 1 diabetes compared with unvaccinated children.
41%. That's huge.
It's a lot, yeah.
Yeah.
What is, okay, stop a moment and just for the ignorant me, tell me what you think that means.
Could mean a couple of things. Either that the unvaccinated children are somehow different from the ones who are getting vaccinated, that there's some selection bias going on here, or that the rotavirus vaccine is preventing infection with something that causes the autoimmune response that leads to type 1 diabetes.
Isn't that worth pursuing, don't you think?
Yeah.
Sure.
Has anybody written about this scientifically?
Well, this study just
This is
came out.
Right.
No,
So
really?
in fact, there's two studies. There's a study of Australian children, and then there's this study of U.S. children. So one was in January of this year, and one was in June. So
And the
scientific
rotavirus
reports
vaccine's only been around for a little while.
Right.
It has. So it might be reason to look and see if there's any epitopes that are similar between rotavirus and in the type 1 diabetes situation.
Is there, in the vaccinated groups of the world, a trend towards reduction in type 1 diabetes as a result?
Well, this is what these
this is
studies
about.
But the insurance company says that, but I mean, what's the scientific?
No, they use insurance data.
Insurance data.
So what they do
Health
now that everything in developed countries is going over to electronic medical records, there are these immense databases of medical records that can now be analyzed in lots of ways, and there are relatively secure ways of doing this so that the data don't get breached. They'll probably be breached some other way, but
right. Facebook will buy them all.
Allow you to do these massive studies where you can look at millions of records at once and algorithmically process them and calculate statistics on them.
So here's the numbers, Dickson.
Yes, please.
12 cases per 100,000 people in the fully vaccinated children. 12 cases per 100,000 per year. Versus in the unvaccinated group, almost 21 per 100,000.
That's incredible. That's a huge difference.
There's no benefit for partially vaccinated kids, those who didn't complete the full number of doses.
Which is what, 3?
Huh. That's interesting.
3 doses, I think. I can't remember.
Wow.
Yeah.
That's quite amazing. And
so if this is not some confounding effect, it's definitely interesting to understand the mechanism, right?
So, sure.
Yeah.
What about in areas where there's a high transmission rate of rotavirus? And so almost every child has gotten that virus as an infection.
Yeah.
What's the rate of diabetes in that group?
I know.
It's like most of the
question.
tropics. Most of the tropics, right?
So this is interesting because I'm not sure what rotavirus vaccines are being used here. There are a couple of different ones, right? They're all infectious. Many of them are infectious.
So they're an attenuated
They're
viral.
attenuated. They're reassortants with animal rotaviruses, so they're not pathogenic, but they have the major antigenic proteins of the human viruses. And so something is different enough so that you're not getting diabetes.
Wow.
So as you said, Brianne, if it's a cross-reactive peptide, then it must be in one of the non-human viral proteins.
Right.
Yeah, that could be true.
Because it's got the same protein as the human viruses.
Yeah, because rotavirus infection doesn't seem to protect as far as we know.
Right, so that makes sense. So you'd want to go ahead and look at those non-human segments and see what the epitopes might be.
And just to bring people up to speed who might not be familiar with type 1 diabetes, this is distinct from type 2 diabetes, which is also called adult onset diabetes, although people are increasingly developing at young ages. Type 1 diabetes is an autoimmune condition that is believed to be caused after viral infection with something that has an epitope that's close to epitopes found normally that cause the body to attack.
Yeah.
Go ahead.
Yes. So they're insulin
the islets of
epitopes.
Langerhans. Yeah. So they're so then the immune system attacks the islets of Langerhans that produce the insulin and just destroys them, and then these people can't produce insulin, and so they develop type 1 diabetes.
Stay tuned.
Yeah,
exactly.
So if you're immunizing against something that prevents that from happening, then
cool.
that's great.
Pictures at 11.
Cool. What did you say?
Pictures at 11.
What is that, a news thing?
Yeah.
Yeah.
What does it mean, pictures at 11?
They're going to show you
we'll have
fuller
footage
story.
of it at the 11 o'clock.
Exactly right. Exactly right. So we have a diabetes center here at Columbia. I'm sure a lot of other places do also. And I wonder what they're thinking about this.
We'll see. All right, we have two more. Kathy, can you take the next one, please?
Sure. Martin writes, Dear Professors, having just attended the 2019 annual Gold Medal Lecture for the Institute of Mathematics and its applications this evening, I felt I had to write to you to suggest a future guest to meet online or in person when she is next in the USA, or you, Vincent, are over in Europe. Her name is Professor Ryden Twarok from the University of York, England. Here is the abstract of her lecture given tonight at the Royal Society, London, as an indicator of how interesting her subjects, geometry and symmetry, are in trying to understand how viruses build their proteins and how she is working with scientists, some of whom reside and work in the USA, to help combat viral disease. IMA Gold Medal Lecture 2019, Geometry as a Weapon in the Fight Against Viral Disease, to be given by Professor Ryden Twarok, University of York, winner of the 2018 IMA Gold Medal. Abstract. The protein containers encapsulating the genetic material of viruses are remarkable examples of icosahedral symmetry in biology. Professor Twarok will demonstrate how techniques from group, graph, and tiling theory have enabled her and her team to solve long-standing puzzles regarding the structures and assembly of these viral Trojan horses. In particular, the concept of Hamiltonian paths has been instrumental in the discovery of an assembly mechanism that occurs in many viral families, such as the picornaviruses that include the common cold virus. Using Gillespie models of viral evolution in the context of a viral infection, Professor Twarok will demonstrate how these insights into virus structure open up novel opportunities for antiviral therapy. She will also show how her recent classification of virus structure based on Kepler's Archimedean lattices has revealed a design principle for icosahedral architectures that solves open problems in structural virology and suggests new avenues in our fight against viral disease. There will be an opportunity after the lecture to experience the models of viral geometry in action in a virtual reality environment.
Cool.
Having told her about TWiV, she would be only too pleased to communicate with you to see if an episode might be put together to further the education of us all in your subject, and how mathematicians do get involved in combating the cold virus, for example. Those 3D AR headset demonstrations mentioned above were really good, letting you appear to be inside a capsid looking out.
Ooh,
love it.
That's fun.
cool.
And then he provides her contact information. Very best wishes to you all, Martin. A TWiV Patreon
love
contributor.
to have her on the show to explain to us what Hamiltonian paths,
Yes, we would like to know that.
Yes, I would like to hear that.
This sounds really interesting.
Thank you, Martin, for doing that, and also for being a contributor on Patreon.
Yes.
So I looked up Twarok's, and she definitely has papers. There's most recent one is in the Annual Review of Biophysics, RNA-mediated virus assembly. And she talks about packaging sequences, dispersed RNA packaging signals.
Cool.
So Hamiltonian paths is one of the keywords in that paper. So
and Gillespie algorithms.
I don't know what either of those are, but we will.
She can explain.
I will explore. Yes, and we will try.
She doesn't have to. She can share you a picture. Pictures at 11.
Pictures at 11. All right, one more. Brianne, did you take that last one?
Sure. Niraj writes, Dear Dr. Racaniello, first and foremost, heartiest congratulations on a very well-deserved education award at ASM. I think everyone on the Twix podcasts would agree that without your passion and commitment, this enterprise wouldn't have grown so big, popular, and resourceful. Too bad I missed the event as I was at ASM the previous day, but did have fun engaging in a fireside chat with Dr. Swanson of TWiM.
By the way, they have these things called fireside chats at ASM meetings now. Just a little, you sit on little comfy chairs with 20 people in a circle and you talk, and they actually have a fake fireplace. It's this digital thing which flames, and it's not warm though.
It's kind of hokey.
It's very. Sorry, go ahead.
Next, I wanted to applaud the terrific interview on polio with Dr. Oshinsky. I really enjoyed the exchange, and I think interviews like these should become more mainstream and part of our everyday dialogue on the importance and need of getting our kids vaccinated. What's happening with measles, especially in states like Texas, is horrible, and discussion like these really inform the common person about the importance and necessity of vaccines, even when the disease burden currently might be well under control. Coming from India, I can certainly vouch for how debilitating polio can be if left unvaccinated against. Sometimes I wonder whether maybe the reason why anti-vaxxers have such a stranglehold on public perception is because they or their near and dear ones have never witnessed the lifelong disabilities that these serious infections can cause. It's sad that kids get put in harm's way, even when we have one of the best human creations to counter it. Thus, I continue to believe that the only way to get over this is by educating and informing the public about the great good vaccines do, and will continue to do so. If you would not leave your kid in a car seat without straps or drive without a seatbelt yourself, there is absolutely no good reason not to vaccinate and protect, since they are incredibly safe and effective. Therefore, thanks for your diligence and efforts in bringing these merits to the common front, especially in a language that is easy to understand and interpret. Please continue the great effort, and here's to many more wonderful years of podcasting and educating. Best, Niraj.
Sorry to miss you. Niraj is out in the Bay Area. Came to ASM Microbe. Sorry to do that. Yeah, when I was talking to Oshinsky, I thought, this is such a remarkable story. Parents lining up their kids to be immunized because they wanted to protect them against disease, right? Yeah. And I think everyone needs to know that story.
But people didn't line up to buy cars with seat belts. I can recall when they introduced that, and the first car manufacturer was Volvo, by the way. And people resisted it, and they said, how dare you tell me how to behave? And we still have people with motorcycles that don't wear their helmets. There are states that don't have a helmet requirement. So people don't like to be controlled at some level, and maybe that's why these anti-vaxxers are reacting the way they are, because they just don't want people telling them what to do. But that,
yeah.
I mean, I think that's the attitude, and then you can't argue about it. But if you live in a
society, you have to be part of the society.
Tell me all. Yeah, well, tell that to Ted Bundy and those people too. I mean, we have a lot of those people in this country, really.
But it seems like if it's because they want to do things because they want to and not because they're told.
Yeah, that's right.
Then if we tell them about the wonderfulness of vaccines, hopefully they would want them.
They would want to.
They don't trust you because, Brianne, you're a scientist.
Oh, how dare I?
If you were a Scientologist, then they would believe you. Then Tom Cruise could be our spokesperson.
Let's do some picks.
Hey.
Brianne, what do you have for us?
So mine is sort of speaking of the icosahedral structures that we talked about earlier. These are two different resources that I use when talking about icosahedral structures with my students, where you can actually get some models of the capsids of a number of different viruses and fold the icosahedra, and actually see what a T=1 or T=3 or whatever virus looks like with the actual capsid protein monomers drawn out on the capsids. And so whenever I teach about virus structure, I have my students make some of these capsid models, and they find it really helpful in understanding the structures. I like the first one, the color-coded ones where you get to see all sorts of nice viruses. Some of the students really like the second one that I've linked to, which has examples that are more sort of mathematical with different T numbers.
Yeah, I did a science fair once at a middle school, I think, or an elementary school, and I brought these. I printed them out and let them make their own. They could take a virus home. They like it.
Yeah, they like it a lot.
Is this a form of origami?
It's just
sure.
Like, is it really? Just,
I mean, it's paper folding.
Paper folding, is that origami?
It'd be really simple, I guess.
It is.
Yeah.
It is.
Okay. Yeah, it's very cool.
These are really cool. I already set up an email to send them to myself for teaching. It's good.
Yeah, they're really useful in class.
Good. Thank you. Alan, what do you have?
I have a book that I picked up a little while ago, mainly for my daughter, but I've also found it really interesting. This is The Law's Guide to Nature Drawing and Journaling, and I will be the first to say that I suck at drawing. So this is not, and I have not taken up rigorously, nature journaling. But what I loved about this is this guy, John Weir Laws, who is not related to John Weir, except just the middle name was inspired by that. He's a naturalist and an artist, and he goes around teaching this sort of stuff, and he wrote this book. And his approach to observing nature is just wonderful. It's a whole philosophy of just asking questions of nature while observing it and drawing it. That's a big emphasis, is how to draw various things. But he uses that as a way into discussing thinking scientifically. And then how can you investigate these questions? And of course, you can investigate some of them in the library or on the internet, but you can also investigate some of them in your own backyard. If you're studying how chipmunks behave and you observe some unusual behavior, you may be able to try to observe other aspects of it that would test your hypotheses. And it's a really cool, approachable introduction to thinking scientifically. And I recommend it highly to anybody especially anybody who's teaching kids about science or trying to get them more interested in it.
I like on page 7, he says, keep track of your questions on the page as they come to you. So it's closed ball.
Yes.
Does this restrict pollinators? Who is the primary pollinator? So, yeah, it's really cool.
Yeah.
And so what have you done, Alan, with this so far?
I've just, I took a little couple of pieces of paper and with me on a hike and did a little sketching here and there, but I haven't really implemented any of it. And as I say, kind of, try to get my daughter interested in it. She's mainly into cartooning for her drawings. But
Now, your grandfather was an entomologist, right?
He was, yes.
Was he a good drawer?
He I think, did a few little drawings, but I don't.
Because a lot of them are. I mean,
yeah, I didn't. That wasn't in any of the records that I inherited from him. He was mainly doing medical entomology, so a lot of micrographs.
Right.
It's a hard word, drawer.
Drawer.
That's true.
Draft.
Kemmerer, Wyoming.
Part of a cabinet is a drawer.
Drawer. It's hard, right? Drawer. Open the drawer.
Illustrator. Illustrator.
That's why people in New Jersey say draw.
Right.
Because they can't pronounce it. Dixon, what do you have for us?
Well, I mean, I have something that I picked because I know that Rich would have if he had been here, so I got there ahead of him a little bit. This is the 50th anniversary of the landing of 2 human beings on another planetoid, the moon. And there are lots of people out there that are anti-vaxxers that also believe that this didn't happen. And the documentary films that they've been showing on television to celebrate the July, I think it was the 19th, I'm not sure, Alan, but it was something close to today's date. July 19th, was when Neil Armstrong and Buzz Aldrin stepped out of their little lunar module and stepped onto the moon. What a risk they took to just do that. I mean, did you have a feeling of how soft or hard the surface of the moon was? And I remember watching all of that with something like 2 billion people that were glued to television sets around the world, including the Pope. They showed this documentary on television recently of NASA's documentary film, and you can get that on this website, of just everybody was just united as a species behind a single event. And that was hopefully the beginning of tolerance and understanding and cooperation and all these other things, which of course it hasn't happened. What they expected to happen as the result of this has not occurred. I don't know why, because this is such a remarkable thing. There was a woman recently once quoted, a woman scientist, that says, when someone tells me that it can't be done, I remind them that there are human footprints on the moon. And there's nothing more challenging to somebody who is negatively inclined than to listen to that story. And that's why there are people out there that don't believe it happened.
And this effort produced what is, to me, the single most astonishing image ever produced, which is the photograph called
The Rising of the Earthrise. That's right. Yeah. That's right. And I think the second one is Neil Armstrong's footprint.
But of course, it was all done in a soundstage in Hollywood, right?
right.
Leftovers from 1925 Buck Rogers movies.
Right.
It's amazing how...
Neil deGrasse Tyson had a good article about that. I think he was the one who did this, that basically concluded the technology to land on the moon did exist in 1969, but the technology to fake it didn't.
That's right. Exactly.
Like our special effects were just not that good, but our rocketry was.
And
we had good claymation.
Watching the close-ups of the construction of some of these things like the lunar module, you can see that it was crudely done compared to what we could do today, for instance. And we're about to go back, I guess. I mean, I think
maybe
the money is there, and I don't know. But that's the other thing that happened after this too. After the celebrations all died down and everything else, people said, well, was it worth it? And that's an open-ended question still.
Yeah. Sure, definitely.
Was it worth it?
Yeah, for sure. A lot of good things came out.
We wanted to beat the Russians, and it turns out that...
No, but the tech is great.
Yeah, the tech is fabulous, and the spin-offs are amazing.
Just very cool. That the fact, just the fact that we were there is very cool.
Question.
But all the tech that spun off and the curiosity, that's all what it's about.
And what's even more remarkable, I think, is the fact that now the space race is between private enterprise.
Yeah.
Richard Branson.
Yeah, they want to make money.
Right. Or, I guess so.
Well, they're just a wash in money, and they have nothing else to spend it on.
Yeah, and they're being, courted by NASA to cooperate and partner with them. And you see what's happening.
NASA is not a wash in money, and Jeff Bezos is.
Exactly.
That's what's going on.
And Jeff Bezos' wife is also
a few private individuals, and that's where.
Tell me about it. That's right.
Kathy, what do you have for us?
I picked another in the map series. This is all 17,069 places on one map, the definitive map of the world's extraordinary sites by Atlas Obscura. So Atlas Obscura catalogs the most unusual, surprising, and amazing places around the world. And so you can start clicking down to your area. And if I do that for Ann Arbor, there are a couple of markers that are one on top of another. And then when you see something pop up, you can click on its link and find it. But for instance, I discovered that of about the 7 or 8 things in Ann Arbor, there are 2 that I hadn't seen yet. One of them is this Food Gatherer's Giant Carrots, 10-foot tall vegetables on the Food Rescue Program site. So, and then it gives a link of where they are, and so I can go check them out sometime.
Dickson, did you know there's these stones on the river here called the Sisyphus stones?
I didn't.
The hordes of precariously balanced stone figures crowd the shores of the Hudson River right near us here.
Oh, wait. No, as you drive down there, of course. Sisyphus.
I didn't know that. Yeah, I drilled down all the way into Manhattan, and that's what I found. That's amazing.
Another one of the ones that's one of my favorite things in Ann Arbor is the Liberty Street Robot Supply and Repair store. And you go in the front of the store, and they've got all these toys and games and things that have to do with robots. But what it really is a front for a program to increase literacy and ability of students to do stuff. So it's just I didn't know that the first couple of times I went there. I just thought it was a robot store. And then there was this curtain going into the back where they do the actual teaching stuff. So
cool. It's very cool.
I don't know how I feel about the fact that 3 of the ones that are quite close to me are all abandoned things.
There you go.
Well,
and I don't know why the Titanic Museum is in Springfield, Massachusetts, but apparently it is.
Right.
Yeah.
All right. My pick is a episode of TWiV that we recorded in San Francisco, Nels and I, with Paul Turner, a virologist from Yale University. And it's all about phage therapy. This is what Paul does from an evolutionary viewpoint. He has a great paper on an amazing story of phage therapy on a gentleman with an infected aortic, what do you call those Dacron things where they stent, and they cured it with phage therapy by just pouring the phage on the sturgeons, just poured it on the stent.
The sturgeons?
The sturgeons. They got fish out of the river.
They had ganoid scales and everything?
It's a great story. The paper just came out. I mean, the sad part, he told us, the day the paper was published, the guy died of a heart attack. So they had cured his infection, which was killing him. He had to go back for repeated antibiotic.
Which infection was it? Chlamydia?
It was a Pseudomonas infection.
Pseudomonas.
But they just poured it on the thing because they couldn't inject.
The operation was a success, but the patient died.
Yeah.
Anyway, he talks in general about phage therapy, and amazing things are happening. Wow. I'm going to just do one listener pick because we have to do a TWiP at 4, and the second is it requires some discussion. So let's put that later. The first one from Anthony is he picks the Global Water Pathogen Project. Mm-hmm. You know about this, Dickson?
I know parts of it. I don't know all of it.
What is it?
Well, there's a huge interest in organisms that are transmitted to humans that induce diarrheal disease. Diarrheal disease happens to be the upper respiratory, and diarrheal disease kills more children than any other combination of diseases.
Mission is to be a resource on water pathogens, which will guide goals for sanitation and achieving safe water around the world.
That's right.
Okay. And so Anthony says, I see this as important for 2 reasons. 1, the world is running out of fresh water. If the choice is dying of thirst or drinking contaminated water, there is no choice.
Right.
And 2, Ewald's hypothesis is that waterborne pathogens will tend to increase in virulence. Waterpathogens.org.
Yeah. Well, we just mentioned one of them, rotavirus, so we're right on target.
All right, that is TWiV 556. You can find the show notes at microbe.tv/TWiV. You can find the episodes on your podcast player on your phone, tablet. Please subscribe to TWiV so that you will automatically receive every episode. There's no additional charge for that service. And we know how many people are listening, which is important for our fundraising efforts. And speaking of fundraising efforts, please go to microbe.tv/contribute and donate $1 a month for our cause. You get not only TWiV, but TWiP, TWIM, TWiVo, and Immune. Of course, questions and comments, TWiV at microbe.tv. Dickson Despommier has returned.
From the dead.
Racaniello.org and the Living River. Is that a .org also?
I think so.
Living River. Thank you, Dickson.
You're welcome.
Good to have you back.
Yeah, it's fun to be back.
Brianne Barker's over at Drew University on Twitter, BioProf Barker. Thank you, Brianne.
Thank you. It was great to be back.
Kathy Spindler's at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove's turbidplaque.com. Twitter, Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.